It's a strange thing to think about. Inside almost every cell in your body is a complete instruction manual for how to build you. A blueprint, a cookbook, a library, whatever you want to call it. It's there. This manual is written in a language that is billions of years old. And it's stored on a molecule so small you need a powerful microscope just to get a hint of it. That molecule is DNA. The amount of information packed into this tiny space is hard to wrap your head around. If you were to print out the full DNA code from a single human cell, it would fill thousands of books. All of that is crammed into a space smaller than a speck of dust. How is that possible? How does DNA store so much information? It's not one single trick. It's a series of brilliant solutions layered on top of each other. Wow. It's about the alphabet it uses. It's about the way it's physically packed. And it's about the incredibly clever way the language is written and read. There's the text itself, and then there's the grammar, the punctuation, and the notes in the margin that tell you how to read the text. Let's start with the absolute basics, the alphabet. When we store information, we use an alphabet. The English alphabet has 26 letters. Computers use a binary alphabet with just two characters, 0 and 1. The DNA has its own alphabet. It has 4 letters. That's it, just 4. Their names are adenine, guanine, cytosine, and thymine. We just call them A, G, C, and T. These 4 letters, these chemical bases, are strung together in a long, long chain, one after another. A T, T, G, C, C, A, G, T, C, and so on. This sequence of letters is the code. That's where the information lives. A different sequence of letters means a different instruction. Just like the order of letters in a sentence gives it its meaning. Dogbit Man is very different from Manbit Dog. The letters are the same, but the order changes everything. Now, a 4-letter alphabet might not sound very impressive. We have 26, but it's surprisingly powerful. Think about computers. They build our entire digital world with just 2 digits, 0 and 1. With just those 2 options, you can represent any number, any word, any picture, any movie. You just need a long enough string of them. DNA does the same thing, but with 4 options instead of 2. This gives it a bit more expressive power for the same length, but the letters themselves are only part of the story. The physical structure of DNA is key. DNA isn't a single string of letters, it's 2 strings. They run parallel to each other, connected in the middle by the letters. This is the famous double helix. It looks like a twisted ladder. The 2 long backbones of the ladder are made of sugar and phosphate. The rungs of the ladder are made of the letters, and here's the first clever trick. The letters don't pair up randomly. A always pairs with T on the opposite string, and C always pairs with G always. This is called complementary base pairing. It's a strict rule. If you have an A on one side of the ladder, you know there's a T on the other. If you have a G, its partner is a C. This rule is a masterstroke of design. It does 2 critical things for information storage. First, it creates stability. The bonds between the pairs hold the 2 strands together, protecting the code written on the inside. Second, it's a built-in backup and copy mechanism. If you have one strand of DNA, you can perfectly recreate the other. You just follow the pairing rule. A gets a T, C gets a G, and so on. When a cell divides, it unzips the DNA ladder down the middle. Each half then serves as a template to build a new partner strand. The result is 2 perfect copies of the original DNA molecule. The information is preserved and passed on. It's an incredibly simple and robust way to copy data. So we have a 4-letter alphabet and a 2-stranded structure that acts as a backup system. That's the foundation, but that doesn't explain how you fit thousands of books worth of this code into a tiny cell nucleus. The next part of the answer is sheer scale and compression. The human genome, the complete set of DNA in a cell, is about 3 billion letters long. 3 billion pairs of A, T, C, and G. To get a sense of that number, imagine it was a book. A typical book might have about 500 words per page and around 2,500 characters. If you printed the human genome in books of that size, you would need a library of thousands of them. All of that information is on a single molecule. The total length of DNA in one human cell, if you were to stretch it out into a single thin thread, would be about 2 meters long, or 2 meters. In a cell nucleus, that is about 6 micrometers across. A micrometer is a millionth of a meter. This is like stuffing 40 kilometers of fine thread into a tennis ball. How on earth does it fit? The answer is packaging. The DNA is compressed in an absolutely epic way. The cell uses a system of spools. The DNA thread is wrapped around tiny proteins called histones. It looks like thread on a spool. This little bundle of DNA wrapped around a histone spool is called a nucleosome. But it doesn't stop there. This string of spools, which looks like beads on a string, is then coiled up. tightly, and then that coil is coiled up again, and that thicker coil is looped and folded and scaffolded into a dense, complex structure. This highly organized supercoiled structure is what we call a chromosome. This multi-level packaging is incredibly efficient. It takes that 2-meter long string and squishes it down small enough to fit inside the cell's nucleus. It's a masterpiece of data compression, but it's not just about cramming it in there. This packaging also serves another purpose. It helps control which information gets read. Parts of the DNA that are tightly packed away are turned off. They are inaccessible. Parts that are more loosely packed are available to be read, so the physical storage method is also a way of managing the data. It's like putting some books in deep storage in the basement while keeping the ones you need right now on your desk. So, we have the letters, the structure, and the compression, but a string of 3 billion letters, even if it's stored efficiently, is useless if you don't know what it means. What does the code actually say? How do you read it? This brings us to the language itself. The DNA code is the master blueprint, but it doesn't do any work itself. It holds the instructions. The workers in the cell are proteins. Proteins are tiny machines that do everything. They digest your food, carry oxygen in your blood, contract your muscles, and fight off infections. They are the bricks and mortar and the construction workers of your body. The DNA's job is to tell the cell how to build every single one of those proteins. A specific instruction for building one protein is called a gene. You can think of a gene as a single recipe in the giant DNA cookbook. One gene has the recipe for the protein that makes your eyes brown. Another has the recipe for the protein that digests milk. The human genome contains about 20,000 of these protein-coding genes. So how does the cell read a gene and build a protein? It doesn't read the master DNA cookbook directly. That's too precious. It stays protected in the nucleus. Instead, the cell makes a temporary copy of the recipe it needs. This process is called transcription. A special protein machine moves along the DNA, finds the start of the gene it needs to copy, and unzips that small section of the double helix. It then reads one of the strands and builds a matching single-stranded copy. This copy is not made of DNA, but a very similar molecule called RNA. This RNA copy is like a handwritten note of the recipe. It's a messenger. It's called messenger RNA or mRNA. Once the copy is made, it detaches from the DNA, and the DNA zips itself back up safe and sound. The mRNA messenger then travels out of the nucleus and into the main body of the cell. Out in the cell, another machine called a ribosome latches onto the mRNA copy. The ribosome is the chef. It reads the recipe. But how does it read the AUCG letters of the RNA? In RNA, the letter T is replaced by a similar letter called U. It reads them in groups of 3. This is the key to the genetic language. 3 letters of RNA form a word called a codon. For example, AUG is a codon. CCG is another codon. Each 3-letter codon corresponds to one specific ingredient. The ingredients for proteins are called amino acids. There are about 20 different amino acids that are used to build proteins in humans. The ribosome moves along the mRNA, reading one codon at a time. The UG means start here and add the amino acid methionine. CCG means add proline. GCU means add alanine. The ribosome reads the codons one by one and grabs the corresponding amino acid, linking them together into a chain. It keeps going, adding one amino acid after another, following the sequence of codons on the RNA. Finally, it reaches a stop codon, a 3-letter word that says end of recipe. The ribosome then lets go, and you have a newly built protein, a chain of amino acids, all folded up and ready to do its job. This system is incredibly elegant. With a 4-letter alphabet read in 3-letter words, you get 64 possible combinations, 4 times 4 times 4. This is more than enough to code for the 20 different amino acids. In fact, there's some redundancy built in. Several different codons can specify the same amino acid. This is a good thing. It means a small typo, a single letter mutation in the DNA might not change the final protein at all. It makes the system more robust, so the information is stored as genes, which are recipes for proteins. And the language of those recipes is based on 3-letter words. This is how the raw sequence of letters is translated into the functional machinery of life. But if you look at the human genome, something strange appears. Those 20,000 genes, the recipes for all our proteins, only make up about 2% of our DNA. 2%. What about the other 98%? For a long time, scientists were stumped. They called it junk DNA, assuming it was just evolutionary baggage that didn't do anything. We now know how wrong that was. That other 98% is where some of the most complex information is stored. It's not junk at all. It's the operating system. It's the regulatory network. It's the grammar and punctuation that controls the 2% of the code that we call genes. Think about the cookbook analogy again. A cookbook isn't just a list of recipes. It has a table of contents, it has an index, each recipe has a title, a list of ingredients, and step-by-step instructions. It might have notes like, serves for, or best for a cold winter day. It tells you how long to cook something and at what temperature. The junk DNA is all of that. It's full of switches, dials, and instructions about the genes. These are short sequences of DNA that don't code for proteins themselves, but they control when, where, and how much of a protein gets made. A section of DNA before a gene, called a promoter, acts like an on switch. Special proteins have to bind to this promoter region before the gene can even be copied into RNA. Other sections of DNA, called enhancers, can be located far away from the gene, but they act like volume dials. When proteins bind to these enhancer regions, they can dramatically ramp up the production of a particular protein. Other regions, called silencers, do the opposite. They act like off switches, shutting a gene down completely. This regulatory information is what makes a human, and not just a blob of cells. Every cell in your body has the same DNA cookbook. A skin cell has the recipe for a neuron. A liver cell has the recipe for a muscle cell, but a skin cell only uses the skin cell recipes. A liver cell only uses the liver cell recipes. How? The regulatory DNA, the switches and dials, are set differently in each cell type. They ensure that only the correct genes are turned on at the correct time. This adds a whole new layer of information density to the genome. The information isn't just in the sequence of the genes themselves. It's in the sequence of the vast non-coding regions that surround them. The meaning of the code depends on its context. The same gene can be used in different ways in different parts of the body at different times in your life, all thanks to this complex regulatory network written into the other 98% of your DNA. And it gets even more complex. There's another layer of information on top of the DNA sequence itself. This is called epigenetics. The prefix epi means above or on top of. Epigenetic information is like a series of sticky notes and highlights attached to the DNA cookbook. These marks don't change the letters of the DNA sequence itself, they don't change the recipes. But they do change how the recipes are read. Chemical tags can be attached to the DNA or to the histone proteins it's wrapped around. One type of tag, called a methyl group, can be attached to the DNA itself. This often acts like a do not read sign, causing the DNA in that area to be packed more tightly and effectively silencing the genes there. Other tags can be added to the tails of the histone spools. These can either cause the DNA to be packed more tightly, turning genes off, or more loosely turning genes on. This epigenetic layer is incredibly dynamic. It can change based on your environment, your diet, your stress levels. It's how our bodies adapt. For example, during a famine, the epigenetic marks on the DNA of a developing fetus might change to program its metabolism to be extremely efficient at storing fat. This is useful for survival in a low-food environment. So now we have multiple layers of information. We have the raw digital code of the ATCG sequence. We have the regulatory code in the non-gene regions that act as switches and dials. And we have the epigenetic code of chemical tags that provides another layer of control. Each layer builds on the one below it, creating a system of staggering complexity and information density. Let's put this all in perspective by comparing DNA to our own technology. The best hard drives we can build store information as magnetic bits on a platter. It's a two-dimensional system. DNA is a three-dimensional molecule. The information is not just in the sequence, but in the way it folds and packs and interacts with itself. The density is mind-boggling. Scientists have calculated that you could store all the digital data produced in the world in one year basebook, all the movies, all the websites, all the emails, all the cat videos, in about a shoebox full of DNA. A single gram of DNA can theoretically hold hundreds of millions of gigabytes. It is, by an enormous margin, the most efficient and dense information storage system known to exist. And it's robust. The double helix provides a backup. The cell has proofreading proteins that check for errors during DNA copying and repair mistakes. The redundancy in the genetic code provides a buffer against some mutations, and the whole system has been field tested and refined by billions of years of evolution. It's built to last. Our hard drives degrade in decades. DNA recovered from the bones of mammoths and Neanderthals can still be read tens of thousands of years later. So, how does DNA store so much information? It starts with a simple but effective 4-letter digital code. It writes this code out in an incredibly long string, billions of letters long, providing the raw capacity. It uses a brilliant double helix structure that protects the code and makes it easy to copy with a built-in backup. It then compresses this enormous string of code through a masterful system of coiling and packaging, allowing it to fit into a microscopic space. It organizes the code into recipes, called genes, but then devotes the vast majority of its length to a complex regulatory operating system that controls how those recipes are used. And finally, it adds a flexible layer of epigenetic marks on top of everything, allowing the information to be modified and managed in real time in response to the environment. It's not a single thing. It is the combination of the code, the physical medium, the structure, the language, and the layers of control. It's a physical library, a computer program, and an adaptable operating system all rolled into one molecule. It's a system so dense and so elegant that our own information technology is still just scratching the surface of what it can do. The more we learn about it, the more we realize that the answer to how is simpler, and at the same time, more complex than we ever imagined. It's just a string of letters, and it's a universe of meaning.